you a global overview about my thoughts on some limitations of data-driven research and sort of where we're headed in informatics and data science within the team that I oversee at the Center for Biomedical Informatics and IT at the NCI. We'll delve into some details related to the Cancer Research Data Commons, the data repositories are available, and the cloud resources that are available for compute and analytics. And then we'll talk a little bit about future vision. So I think that, just like with any other area of science, data science is just exploding. I know that all of you definitely appreciate that with the work that you're doing. And in my view, there are, some limitations, which you could also view as strengths as well. But I think that we need to be, aware of these things and thinking about these things as we're moving forward, especially at the NCI, as we're building infrastructure to make data available and useful. NCI funded research generates all types of really valuable cancer research data, but many times those data are difficult to find. The analysis of all the multimodal, multi-level data from multiple sources can be very challenging. I know many of you are working on this problem, and I'm glad to hear about that. Most researchers are not bioinformaticians or data scientists, right? If we think about the broad range of disciplines within the cancer research community, just in the United States, if not internationally, most folks are not quantitatively inclined. And so, we feel it's important at the NCI that moving forward with the Cancer Research Data Commons, that we are continuing to sort of push the envelope in making the data available and useful in a way that regardless of your discipline or training level, you have the ability to leverage our data assets. And that we do that by making the data available, queryable, and also providing the availability of analysis tools and platforms. Data right now within the Cancer Research Data Commons, as you will see, are stored in separate data repositories based on data type, but we are in the process of creating a combined portal that will allow you to search across all the repositories and find the data that you want to use. So, we like to think about the Cancer Research Data Commons as a foundational piece to thinking about how do we build a national cancer data ecosystem that allows us to integrate surveillance, patient level data for discovery and analysis. And so we will talk today about what's currently available in the Cancer Research Data Commons, the Genomic Data Commons, the Imaging Data Commons, the Integrated Canine Data Commons, the Proteomic Data Commons, the Cancer Data Service, and then soon to come the Clinical Trials Data Commons. And so these along with the 3 NCI cloud resources make up the current state for the Cancer Research Data Commons at the NCI. And we hope that we are empowering researchers by allowing them access to cancer data through state-of-the-art infrastructure for visualization, analysis, and interoperability between different data sets. We do use a cloud-based computational environment, which allows us a certain amount of flexibility. And, why are we doing this, right? Why is this available? Because we really understand and appreciate the value of data and how it can enable discoveries in the short term and long term. So we want to preserve that value. And we are in the process of making plans for improving ease of our data submission, access, and interoperability within the CRDC. And we hope that all of this, right, accelerates cancer discovery. The CRDC is built upon a data commons framework, so it is reusable and expandable. It has very specific core principles and structures, and the components are modular. And I think some of the really critical components I want to point out to you is that we have a centralized authentication and authorization process to get into different levels of data within the CRDC. We use a central indexing process so that individuals get a very specific unique ID to themselves, even if they have data in multiple repositories, so that their data can be linked together. And then we do have cloud storage and compute available through both Amazon and Google partnerships. We have 3 cloud resources available. They provide you access to specific workspaces just for you and your team, where you can leverage analysis tools and workflows and pipelines, and also the data that is already there within the CRDC. You can therefore bring your own data into your workspace and analyze it with data available in the CRDC, so you don't have to worry about downloading all of the data from the Cancer Research Data Commons. So we have 3 cloud resources available. We have the Institute for Systems Biology Cancer Gateway in the cloud, and it's really great for command line analysis using their BigQuery databases. And we have a lot of specialty databases that require a little extra attention, such as a P53 database and the Middleman database that are available through the ISB cloud. We have the Broadfire cloud, which is really great for running production pipelines. So if you have pipelines that you have put together yourself, that would be a really great place. And then we have the 7 Bridges Cancer Genomics Cloud, which provides a much easier sort of non-technical, non-programming user interface with a lot of really great visual displays. I encourage you to take a look at our website, datacommons.cancer.gov. We also have a training module. All of the cloud resources have training modules and office hours and are really there to help you if you are interested in using them. Just to give you a little idea, high-level overview on statistics and impact of the Cancer Research Data Commons to date. If we look at the specific repositories within the CRDC, the GDC, or the Genomic Data Commons, has been around the longest. It was really born from needing a place to make the Cancer Genome Atlas data publicly available. And so you can see that it has a very large number of users per month and a lot of data there. On over 30 different cancers and about 11,000 individuals. The Proteomic Data Commons is born from the Clinical Proteomic Tumor Analysis Consortium studies, and that's where their data resides. I know some of you have worked on those studies, and so those data are growing as CPTAC grows. The Imaging Data Commons is also growing. It's one of our newer repositories. And I'll tell you a little bit about the validated de-identification pipeline that we're putting in place to make it easier to deposit imaging data there. The Cancer Data Service is a specialized service where data sets that may not fit exactly with the other repositories can come into the CRDC that way and be shared. And I'll tell you a little bit about that as well. And then the Integrated Canine Data Commons is really interesting because it is a network of veterinarians who are running clinical trials in canines and dogs, specifically immuno-oncology trials. And then they do all kinds of sequencing on the dogs, and then they have human data as well to compare that to. The NCI cloud resources are also very highly prescribed and used. You can see some statistics here. And across the Cancer Research Data Commons, the data is heavily used and published on, and studies and collections are released from there. So I want to delve a little deeper into each of the repositories and provide you some really fun new things maybe need to know about each repository. So again, this just gives you a high-level overview of the different repositories and what's available. So if we focus first on the Genomic Data Commons, this just shows you a view of sort of the front page for the data portal. Some important things to point out about the Genomic Data Commons is that they have a standardized pipeline that they use. And so if your data are accepted into the Genomic Data Commons, those data are run through the same standardized pipeline. So then you can compare with any other data set that's already included in the Genomic Data Commons. Here are some big data sets where the genomic data, so this is the DNA sequencing data, is included. You can see, of course, TCGA, but also Genie, the CPTAC genomic data, Target, which is a clinical trial in children with cancer. And, you know, just so you know, some of the things that you can do with their visual interface, you can compare custom cohorts based on clinical data and genomic data. You can also view mutation frequency by cancer type. And so, you know, it allows you to get some high level overview of, you know, whether or not data that you might be interested in could be found in this database, especially if you're interested in looking at a specific type of cancer, a specific group of patients with a particular mutation. It provides you with an oncogrid type view of mutations across individuals where you can put different tracks there based on what other type of clinical data or other mutational data you're interested in. And it also allows you to do Kaplan-Meier survival analysis as well. And, you know, you can download the plots and use them. And so, you know, it's heavily used in folks' research. I think they also are using it as a feasibility type study to see whether or not there would be enough patients within these data sets in the GDC for their own research questions. You can see on the upper left here that the amount of data that's been released by the GDC has steadily been growing over time. And there are over 67 primary sites included and you know, almost 100,000 cases. And it is one of our most heavily used repositories within the CRDC. Delve a little bit further into the Proteomic Data Commons, which is a partnership with the Clinical Proteomics Tumor Analysis Consortium. Again, they also used a harmonized pipeline to look at all of the proteomic data. Just to give you some data metrics, they have 15 disease types from 12 primary sites. It's about 100 studies. They have about 3,000 to 4,000 users per month, and it's international use. And down in the bottom here in these little tiny pictures, you can see that they've had a lot of very high profile papers that have been published through the CPTAC in multiple different cancers. And so what can you find within the PDC? You can find all of these different types of data: whole proteome, phosphoproteome, acetylome, glycoproteome, and ubiquitolome. And there are multiple different types of experiments that have been done, and you can get access to not only the raw data, but also processed data if you're interested. It allows you to explore across the studies, so you can see a summary of all the studies, and then you can filter by various metadata types so that you can look by biospecimen type, whether it's tumor or some other type of specimen, by various clinical variables, by gene, and also by protein names. And then you can also look at the study summary. So if you've done your search and you find some information that you want, you can easily bring up which study those individuals were generated from, so that you have a full understanding of the protocols and experimental design and what was collected on them, so that you can, along with references, so that you can use it further in your research. I think the other thing that's really great about the PDC, it's one of our newer repositories, is that they've really worked hard on interoperability between the different repositories. So they have data in the Proteomic Data Commons, the Genomic Data Commons, and the Imaging Data Commons, because they do capture all of the different types of proteomic data, genomic sequencing data, and imaging data on these individuals that are included in these studies. But in addition, they have assay data available and antibodies available, which I think is really exciting if you are interested in this proteogenomic space. If we move on to the Imaging Data Commons, you know, it uses the DICOM standard. So for any data that are submitted there, the files are converted to DICOM. It includes data now from some very large studies. And it has a very nice user portal that you can use to search the metadata. You can see here on the bottom left how many cases approximately are available by primary site. And so it continues to grow. And you can see the little movie running here on your bottom right. And one of the new features for the Imaging Data Commons is a viewer, an imaging viewer. And so once you have determined sort of your case listing, you can then view the images that are available. So this is just showing you a digital pathology slide sorry, the word is escaping me right now. I can't talk. But it allows you to sort of zoom in and out and see what's going on. This is an H&E slide. And so it has both radiology and pathology images, and it allows you to look further into the imaging annotations as well. The Imaging Data Commons, we hope, is set up in such a way that it will allow for ease of use of the images for AI-based analyses. And so it allows you to do data retrieval. You can do this actually through the Google Cloud platform and the viewer. And then it allows you to pull those images directly into one of the cloud resources or put them in a space that you have for analysis. Images are very large files, and so many times it does require that you use some sort of cloud computing situation or some other high performance computing. Just to tell you a little bit about, which I alluded to before, that we are in the process of validating a cloud-based automated de-identification pipeline for imaging data. And we are doing this in partnership with Deloitte and Google. And we have some very nice preliminary data where we've taken an initial set of almost 2,000 images and been able to de-identify them to up to almost 100% accuracy. And so we are in the process of validating this pipeline on data with real identifiers, because we sort of had some data sets that we used that had fake identifiers on them, and we're getting the contracts put in place to be able to do that so that we can ensure that it really is de-identifying the images in an appropriate way. And then we will launch this and make it available for folks to put even more imaging data in the IDC. Talk a little bit about the integrated canine data commons. And like I said, it includes data from mostly immuno-oncology studies in canines. And it does include whole exome, whole genome, RNA-seq, DNA methylation, and clinical data. Right now, there are about 8 clinical trials worth that are included in there. But, just to give you a little scientific use case, let's say I want to study dogs that have bladder cancer, that have samples in the biobank, so that I can evaluate differential gene expression across different stages of disease. And so I would start from the integrated canine data commons portal, and I would be able to create a manifest that I could directly import into the 7 Bridges Cancer Genomics Cloud. Oh, sorry, clicked too fast. And then it would bring my all of those data into the Cancer Genomics Cloud for me, where I could then analyze those data. The Cancer Data Service is a way to access NCI-funded data that is currently not hosted by other repositories within the CRDC that we've just gone over. All data types are accepted, so it doesn't have to be imaging, genomic, or proteomic. And right now, 2 of the major big NCI studies that we include data from are the Human Tumor Atlas Network, as well as the Childhood Cancer Data Initiative. And so we really are able to take almost any type of data, and we have a FSMA moderate authentication to operate. So it allows us to also take data that may have special considerations for extra protections. And just to give you an idea, we really started in the end of the year in 2020. And this just gives you an idea of some of the data sets that have come in, and you can see that it's, you know, an interesting grouping of data sets that have all different types of data available. And so you can start from the Cancer Data Service website, and then you can use their browser, similar to how you can do with the Integrated Canine Data Commons, and you can upload that manifest to the 7 Bridges Cancer Genomics Cloud. And then you would be able to actually analyze your data from there on the cloud. Again, the other thing to note about when you're using the cloud resources is you have your own dedicated workspace. You identify the people who can get access to that workspace from your team or other collaborators. And then you can also bring other data into that space to analyze along with data that you would pull from the Cancer Research Data Commons. The Clinical Trial Data Commons is coming soon. It's going to share data from NCI funded trials. And so we are in the process of getting all of that together. We'll be starting with some of the precision medicine trials, specifically the MATCH trials. And here are some of the arms of the MATCH trials that have already been published that we are anticipating will come into the Clinical Trials Data Commons soon. So just to talk a little bit about, you know, sort of, okay, great. So you just described all these repositories and the Cancer Data Service and you know, things that are coming. You know, what else are we thinking about that we think are important to make sure that these resources are really available to everyone in the cancer research community? Just to review some of the challenges that we had talked about, which we think are really important. You know, we need as the NCI, you know, heading up this Cancer Research Data Commons, we need to make it as easy as possible to deposit data, to find data, to access data, and to interoperate between multiple different data types. At the same time, we need to make sure that our infrastructure is up to date and robust and follows all the right rules for data sharing. So the CRDC, what is it? I mean, it's a place to explore cancer data. We have all types of cancer data, which I hope maybe you learned something new that you didn't know was there before. But it's not a place for every type of cancer data necessarily, and there are other NIH repositories that might be a better choice. At this time, we really are resourced to take the big, large data sets. We're not really resourced at this time to take data from every R01 grantee, but we're hoping that we'll be able to expand our scope in the future. And so just a grander vision of where we're going within CBIT and Informatics and data science is, you know, we want to be engaging with you, with the extramural community, like I'm giving this talk today, because getting your input is really important for our success of any program. And we do engage intramurally at the NCI as well. We feel it's our responsibility to lower barriers to data submission, data harmonization, data access, search and retrieval, being able to combine data sets across our repositories, and also analyze those data sets in an easy way. At the same time, we need to balance that with how much things cost, right? Making sure that we have the right security in place, because we do have some sensitive data sets. And make sure that we have a sustainable and reusable architecture and a comprehensive plan to make sure that we're storing all of the data and the tools and making them accessible as we move forward. So just to remind everyone about the, you know, our thoughts on the National Cancer Data Ecosystem so that we can integrate data from all of these different sources. And we're hoping to expand the Cancer Research Data Commons in the future to, you know, include other types of data commons. So I just want to kind of put all of this in a little bit of context. And Dr. Chen, thank you so much for the very welcoming introduction. I do brain tumor research, and so I thought maybe I could give you a little example about how some of these big data assets have really been a game changer for brain tumors in terms of how we diagnose them and how we think about them. Unfortunately, for brain tumors, we have a lot of gaps in our knowledge. We have very limited treatment options, which means that we really don't know how to do individualized treatment plans. We need to have a better knowledge of the biology of the disease, especially including the microenvironment and how that impacts the tumor growth and tumor response to treatment. I think one of the big challenges we have is we don't have a risk factor that explains a large number of brain tumors. And maybe this is because we need to be better at measuring risk factors. I think the population-based statistics are really critical to our understanding of burden of the disease in the population. And I'll provide you with some of that information here. And then how do we leverage all these different types of data that are being generated to make new discoveries that could make a difference for brain tumor patients and their families? The Central Brain Tumor Registry of the United States is data that includes all 50 states' cancer registry data so that we can provide accurate and up-to-date statistics at the population level for the United States on brain tumors. Brain tumors come in two big groupings, non-malignant and malignant. You can see that this pie chart is showing you overall for the entire United States, all ages, that non-malignant brain tumors make up about 70% of all brain tumors, malignant about 30%. One unique thing about brain tumors is that with the passing of the National Cancer Act in the early 1970s, that's what established the state level cancer registries and required them to start collecting every newly diagnosed cancer case in the United States. Clearly, non-malignant tumors were not included in that mandate, but in 2004, the Benign Brain Tumor Act was passed that now required a collection at every state level of non-malignant brain tumors. So we now have, a good number of years available of non-malignant to be able to really show, statistics. Probably you guys are familiar with the term glioblastoma. That's the most common type of malignant brain tumor in adults, and some very high profile people around the world in the United States, unfortunately, have succumbed to that disease. Incidence or the proportion of newly diagnosed cases in the United States varies significantly by age. So in children, it's the most common solid tumor diagnosed in children. Obviously, the most common cancer in children are the blood cancers. But you can see here that the distribution of malignant versus non-malignant varies significantly, whether it's in young children or in adults. So in children, the malignant tumors are much more common than the non-malignant tumors, and in adults, vice versa. If we then look at just some very straightforward Kaplan-Meier curves where we've stratified to the different age groups, children 0 to 14 years old at diagnosis, adolescent young adults 15 to 39 at diagnosis, and then adults 40 and older. And we're just showing you here stratified by specific histopathologies that are common in those age groups. I think the thing that's really striking is the glioblastoma line, or the red line, which shows that it doesn't matter what age you are, you unfortunately have poor prognosis. But for the AYA group in the middle and the adults on your right, you can see that those that have an oligodendroglioma have the best survival. And I'll tell you a little bit more of a story about oligodendroglioma and how these big data assets impacted our accurate diagnosis for that tumor. So we've known for quite a while that there are some very specific molecular hallmark features that are really critical for brain tumors. I think we didn't really understand, do we need to incorporate those fully in diagnosis or not, until we learned some very important information from the TCGA studies in both glioblastoma and lower grade glioma. On the left-hand side, this is a Kaplan-Meier curve from the second paper on glioblastoma from the Cancer Genome Atlas Project. It included almost 500 individuals. And I think the striking thing here is the 4 lines that you see together, clustered together, were gene expression-based subtypes of brain tumors, which I think have biological relevance. But for those of you that are used to reading Kaplan-Meier curves, clearly they don't have clinical outcomes association significance here, with the exception of the purple dashed line, which are those individuals who have an IDH mutation. And that is a very small number of glioblastomas that do, but they're now no longer classified as a glioblastoma if they have an IDH mutation. So that's very interesting. The figure on the right is from the lower grade glioma studies from the TCGA. And what we showed here was that in order to accurately diagnose an oligodendroglioma, period, it must have an IDH mutation and 1p and or 19q chromosomal deletions. And so what happened from these studies is we then went on to, it had an impact on the World Health Organization of classification of brain tumors with a change in 2016. And this is what I have highlighted showing you here, where some of these hallmark molecular features are now included in accurate diagnosis for brain tumors. So you must have these features in order to be diagnosed with this type of brain tumor. There was an expansion and a revision in 2021 to the WHO classifications for brain and other CNS tumors, where they did expand on some of these groupings and added in the possibility of additional molecular markers to be added to these hallmark markers. We were able to work with the cancer registry community in the United States to add a new variable so that these really critical hallmark molecular features that are now incorporated into diagnosis for brain and CNS tumors could be collected in the United States. They started collecting those in 2018. So we now have 2 years worth of data available. And it's very interesting to see what are the population level statistics for these different types of molecularly diagnosed or defined brain tumors. So this is definitely a team sport. We have a really amazing team at CBIT I'm very grateful for, as well as the support of the CBIT leadership team, all of our partners throughout the NCI, NIH, and externally, and all of our contractors. Also grateful for my research team and my co-PI for the Central Brain Tumor Registry of the United States, Dr. Quinn Ostrom from Duke, and our founder, the founder and CEO of CB Trust, Kjell Kruchko. And that's where I'm gonna end. And here's my email. And I'm very happy to answer questions. I'm also very happy for anyone to email me if you have additional thoughts or questions, if you think about things after the talk today. Again, thank you so much to Dr. Chen and Dr. Wang for the invitation.
Thanks for the excellent presentation. And I really appreciate you giving so much of your time and expertise to UAB over the last couple of days. Thank you so much.
It's been a UAB week, right?
Yeah. And also the fact that this talk was so, letting us know what kind of resources are available for big data and how to take advantage of some of those resources, and then illustrating how that can be used. As a clinician, I was really impressed with how you're showing how when we had our prior understanding of brain cancer, often was incorrect, probably based more on phenotype rather than genotype, and that you've shown that what we thought was one kind of cancer is actually a different kind of cancer, and that has such great impacts on treatment. So, just wondering how. What are other disease areas that you see that other cancers that there's going to be a bigger focus, or are you just showing the brain cancer as an illustration that's because that's your area of expertise? That
yeah, right.
Oh,
right.
okay.
No. Thank you so much for bringing that up. So I should say that TCGA obviously wasn't just a game changer for brain tumors, right? It was a game changer for a lot of the other cancers that were included in TCGA. I believe they have over 30 different types that are included in those data sets. And CBTAC and the Human Tumor Atlas Network have then expanded more work and generating more different types of data for many of those different types of cancers. I think what's interesting is that glioblastoma was one of the first cancers to be studied in the TCGA, along with ovarian cancer and squamous cell lung cancer were the first 3 that were included. So, that started a long time ago, right? 2008, 2009, 2010, somewhere in there. So, we're a little bit further down the path in terms of knowing what to do with all these data and how we make heads or tails of it. I think in addition, brain tumors don't seem to be maybe as complicated as some other cancer types. And so their mutational burden is lower than a lot of other cancer types. Some of the cancer types, and there have been multiple studies that have been published sort of, if you guys are interested in the pan-cancer atlas studies, are very interesting, where they've looked across all the different cancers that are included in the TCGA and tried to find sort of common things, you know, across the cancers. The tumors that have the highest mutational burden, believe it or not, are those that have a what I call, and this is not a scientific term, a whopping risk factor. Okay, so a melanoma with UV, you know, exposure, a lung cancer with, you know, tobacco use exposure, a bladder cancer with tobacco use, alcohol exposure. Those types of tumors have the highest mutational burden. I'm talking about, you know, DNA based mutations. But I think that the illustration I gave you in brain tumors, if someone else was giving a talk and they were an expert in lung cancer or pancreatic cancer or ovarian cancer, they could tell you a similar story based off of some of these big data sets for those cancers as well. I think one of the big challenges, right, is, and all of you guys will appreciate it because you're all quantitatively inclined, right? So we're always worried about, or I hope you're worried about, study design, sample size, power, right? You know, do you have the sample size and the power to actually answer the question that you have? And so one of the challenges that happens with all of these data that you integrate together is now you're slicing and dicing your, you know, you may start with 200 individuals, but then you find that these 5 people have this profile and these 5 people have this profile. And how do you then make heads or tails of it? And so, you know, I think that these data sets that we have available, I hope will be useful for not just like hypothesis generation, right, where you can go in and sort of visualize and ask questions, but maybe if you have your own patient set that you're studying, you know, could you use these resources for validation? But when you get into all these rare subtypes, you know, it just becomes challenging all around. So I don't know.
I was actually thinking when you mentioned that yes, it seems like it's all moving towards personalized medicine, precision medicine, as you realize that these people you thought were one big group are not. And there's a question here from Chen Huang. Thank you for the great talk and the information. For some analyses, different processing pipelines might lead to different results. Could you comment on how the GDC/PDC/other centers chose pipelines to process and harmonize the data level 2 and above, like mutation calling, annotation, protein identification? Will and how frequent will these pipelines evolve in the near future?
Great question. And I may not be the best person to fully answer in terms of why they made the choices that they did in terms of the different components that they included in the pipelines. For the GDC, I know that they update the pipelines on a pretty regular basis. I think every 6 months or so, they're kind of assessing the pipeline, making sure it's as robust as possible. And when they do update it, they run all the data that they have in the GDC back through the pipeline, right? I know that they use multiple different mutation callers, and I think they then take consensus calls from that, which, by the way, was what the TCGA studies did, right? So I think that they're just sort of following along with that. For the proteomic data commons specifically, you know, that's a little tougher, right? Because for the GDC, the data that they have is going to be exome sequencing, shallow whole genome, or maybe deep whole genome, let's just call it. So that's a lot more straightforward, I think, than remember the slide where I showed you all the different data types that the proteomic data commons has in it. And you can imagine that each of those different data types has to have a separate pipeline. So I know that the WashU team, I'm involved in the glioblastoma studies through CPTAC, and the WashU team under the leadership of Li Ding runs a lot of those pipelines for the proteomic data. And I'm sure she'd be happy to talk to you or someone else on her team. I don't know how they made all those choices. But you know, the idea with any of these commons is that we want the data any of the data that's available in the commons to be used together. And so they are run through common pipelines.
Yeah, thanks for that. And another question from Stephen Barnes. Feel free to enter questions in the chat, or you can also unmute yourself.
If you want me to ask the question, so people who know me will say, well, where's the metabolomics? Where's the metabolome? Because we obviously our systems are driven by small molecules every second of the day, and without them, we won't exist. So I was really pleased when you ended up on the IDH mutation, and also that it actually gave. It was the only one that sort of changed the rules, and it's really the tumors have to have a flow of carbon atoms going into the pathways to build a tumor. So if you can interrupt that process, then you obviously are going to control the ability of the tumor to grow. And so the IDH mutation stops the formation of alpha-ketoglutarate, and that there's two sides to that. It stops alpha-ketoglutarate going to glutamate and glutamine. Those are pretty important things in the brain. But it also means that that the whole cycle is affected, and alpha-ketoglutarate is actually a regulator of many processes that are going on. For instance, we saw alpha-ketoglutarate disappear during puberty in the mammary gland. And this is really important because alpha-ketoglutarate is regulating HIF1 lifetimes. The normal HIF1 lifetime is about 5 minutes. And it depends on two things. One, the availability of oxygen, and the other is the availability of alpha-ketoglutarate. So if you don't have it there, then HIF1 alpha stays around and now goes into the nucleus and initiates a whole program of events that are associated with cancer. So I guess the big question is, when are you going to put the metabolome into your big data sets? Because the metabolome represents a lot more than just what we can discern from the genome. Because if you just look at the amino acids, half the amino acids that make up the proteins that keep us going have to come from our diet. So the diet brings in totally other genomes, as in does, of course, the microbiome. So I think this is really important if you want to get to the bottom of individualizing people's responses to the cancer they're getting. So what's the plan?
So in the Clinical Proteomic Tumor Analysis Consortium, CPTAC, they are now generating metabolomic, lipidomic, and maybe microbiome, I think, data on some of those data sets. I think what happened there, at least with GBM, is they collected an initial brand new set, prospectively recruited and consented set of GBM patients that aren't in any of these other data sets, right? And published a paper on that in Cell. And now they're doing a validation study, and they have expanded the omic types that they're generating to metabolomics, lipidomics, and I think microbiome, but I might be wrong on that.
Yeah, well,
so the data will be available eventually in the proteomic data commons.
Okay, well, this is good because next week I'm chairing a workshop on precision medicine at the Metabolomics of North America Association in Edmonton in Canada. So we're actually, there's a working group trying to see how we can get metabolomics data pulled together, obviously new technologies that speed the whole thing up. We'll be talking about dried blood spots as a way of facilitating and making all of this much more regular. But the metabolome is so dynamic, and it really depends where you when you sample it too, as to the meaningfulness of the data.
Definitely.
It's huge.
Thanks for your question.
Thank you.
Well, Joe, I have a series of connected questions since we don't have you here very often. And I'd like to maybe either provide you some feedback or just our thoughts. I cannot really find the answers on the NCI's the website. So first of all, we really like what NCI have done on the cloud, and it's really world leading. And if it's not for the center, and we wouldn't even see what the cloud can do, right? So you're aggregating a lot of data funded through NCI and adding functionalities and tools and data set to it, making sure that people can learn. But having said that, and it was a little bit of confusing, at least to me, what this future picture would look like. You clearly shows the picture that is going to be very comprehensive, that will actually really entice a lot of the traditional work done at the university or community levels, done at the cloud level. There's a clear benefit. But do you see that it's going to be a one cloud that rules them all, or an ecosystem? Because I'm looking at everything that NIH has, is NIH cannot even agree on a single cloud, right? There's N3C and there's this and all of us, and then they are all on different cloud, and that you're using vendor like Google and 7 Bridges, but we all know that Amazon also have very good machine learning algorithm that Google doesn't have, and Oracle have very good cloud, and Microsoft automatic machine learning. And just because Google and Amazon and Broad become supported funders, doesn't really give them the dominance on the providing of the tools. So if you do believe that there's going to be an ecosystem, then don't you think that the small players need to deserve like some non-competitive funding to actually use it for crash and burn type of applications, and not just for the final stage of development, deployment type of applications? I know that there's a Stripe program, but Stripe program only provides 6 to 10% discount. It does not provide the errors and mistake that some of our students can make in using the public cloud. So that's a lot, but I put some notes in the chat box.
Yeah, sure. So let me try to maybe peel off the layers, Jake, if I could.
Yeah.
So first of all, CBIT does not grant grants, so I can't really speak to the funding aspect. What I can speak to is some of the things that we are thinking about and working on to make the Cancer Research Data Commons more user-friendly, more accessible, and that type of thing. So we are in the process of having a contractor, an external contractor, look under the hood, right, at all the finances, all the components of the Cancer Research Data Commons, and give us some feedback on how to make sure that we are sustainable moving forward, balancing whatever choices we're making for the CRDC with making sure that we are complying with making things findable, accessible, interoperable, and reusable, right? And the way the CRDC is now, I think will be different in a couple of years, how it will be. And so what we are in the process of doing, however, while we're waiting for these sustainability study results and to make some decisions about that, what we're going to do with all that information, is we are putting in place a cancer data aggregator, which will be an API portal on the front of CRDC that will allow you to easily search across CRDC and assemble the data set that you want, instead of having to go to each repository and find data right now, which is way too burdensome, honestly. So hopefully that will help. In addition, we are putting in place a data coordinating center, which will allow for very clear we hope, easy to use way for folks to deposit their data in the Cancer Research Data Commons, so that we really can expand the ecosystem of data that's available. For the cloud resources, we do have 3, right? And so it can be kind of confusing to figure out which one you use. What I will say about that is that obviously taking, a look under the hood and taking a hard look at what we're doing with those cloud resources and what they're bringing to the table is part of the sustainability study. So, stay tuned. I don't know yet what's gonna happen there. What I can say is, and I don't know if you guys have been taking advantage of this, but for each of the different cloud resources, you get a certain number of free credits with them. And, I think it's 250 or 300, something like that. I know for sure for the ISB cloud, and I think the other clouds, you can also apply to get additional credits from them. So if you do have students or faculty who are interested in using the cloud resources, again, so you don't have to download petabytes of data, right, and then have somewhere to put it. I would make sure that when you are communicating with the cloud resource to set up your workspace, that you are clear that you're getting the free credits first, right? That you guys are making sure that you're killing all of your jobs before you get out, because sometimes you can leave them running and they run all night long, or they run for 2 days before you log in again, and then you burn up all of your free credits, right? And then also ask them specifically, tell them about the project that you're working on, the importance of the project, where you're going with the project, and is there a way for you to apply to get additional free credits from them? I don't know if that helps at all.
Yes, yeah, well, thank you.
Sure.
We only have 2 minutes, but I'm pretty sure.
Sorry, I haven't checked the chat.
I'd like to ask a quick question. First, Jim Cimino, Director of the Informatics Institute. So first, I want to say thank you very much for this very comprehensive talk. This is really very helpful. I do have a question, and I know that CBIT doesn't do extramural funding. I used to be an NIHer at one point in intramural program, but I wonder, a lot of cancer research depends, of course, on data that comes from electronic health records, and I'm wondering whether CBIT is happy with the kind of data they're getting, or whether they would like to see changes in electronic health record design or implementation to improve the quality and quantity and breadth of data that comes from electronic health records that's used in research, but whether it's, sort of data sets on, cohorts, or it's data collected in the process of research that, both of those, a lot of them, the latter also come through electronic health records sometimes. So I'm wondering if CBIT's thought about that at all.
Well, I think we talk about it all the time, right? Who doesn't, because, but I'm sure all of you greatly appreciate the messiness of electronic health record data, right? And so if we're going to be able to leverage it from multiple different sources, there has to be a way for us to work off of some accepted data standard, so that the data are coming in per that standard, right? Per that standard data model. I think what N3C has done with the COVID data from the CTSAs is really amazing, right? But the way that they did that is a very federated model, right? They pushed out to the sites what the sites would need to do to OMOP. It's just interesting to me, we use that as a verb now, but okay, OMOP their data, right? Put it into this standard common data model, and then have the ability to send queries across all of the different data sites. So it's something we've been talking about how will we do such a thing for cancer, but it's a complicated problem. I will say that the Childhood Cancer Data Initiative is tackling this problem, Jim, but for childhood cancers only, right? But I think the important thing there is that they're trying to work it out, right? So that we would have these mechanisms to get electronic health record data and standardize it, so that it can be widely used and easily queried. If they can figure that out for childhood cancers, we should be able to then extend it for all the rest of the cancers, in other groups.
Well, just to say that the problem, I think they've solved the problem with standardization for the data that are there. But N3C, you can't tell from N3C data who's really sick and who's mildly sick. It's very difficult to tell that without a lot of phenotyping and 20 questions. You can't tell who lived and who died because the patient might have been discharged and died at another facility. So there's a lot of problems with the quality of the data in the EHR. So we can standardize all we want, but if it's not good data to begin with, we have a problem. So I don't want to belabor it, but it's something to think about, is that it's more than just getting the data out of the EHRs. It's getting the data into the EHRs that I think is something that's not being addressed. And, NCI is the only institute besides the NLM, I think, that pays attention to the research issues around that.
Well, if I might just flip it on its head a little bit on your statement, maybe it's just not feasible to get all the data that we would want into the EHR. Maybe we have to strategize about what other data sets need to be linked together, right? So,
Okay.
there's highly curated cancer registry data at every state's level. It has its caveats, right? There's the National Death Index data. It has its caveats, right? But there are many other population-based data sets that might be really helpful, especially I think at the outcomes level, because one of the things I think this is what you were saying, Jim, is that it's just the outcomes aren't there in the EHR data, right? Because folks bounce around to different health systems. And many times you can't get the comprehensive view on an individual by only looking at one health system's, electronic health record. I know N3C is using some of the privacy preserving record linkage algorithms,
Right.
to try to deduplicate, or if there's an individual who was seen at two different centers, they can somehow get the data together.
Right. That's right.
From them. But it's a complicated problem for sure. And I do think the childhood cancer, it's not that I think, I know that the Childhood Cancer Data Initiative is trying to tackle some of these problems, but maybe it just hasn't gotten as much attention because it's only in the childhood cancers. And I don't want to minimize the importance of studying childhood cancers. Obviously, it's very important. But I think it was like, this was what was easy to sort of bite off first to try to figure out some of these processes and deal with some of the challenges that you brought up.
Okay. Thank you. And thanks again for speaking to our group.
My pleasure. I'm just thrilled. Thank you so much for
Okay.
Well, we want to thank you again and the virtual classes, and hopefully we'll keep in touch and thank you. Yeah.
Well, I hope to be able to see you guys in person. I'll be moving to Atlanta soon, so I'll just, I think, a couple hours from you guys, right?
Yep, that's right.
Yeah.
All right.
Okay, great.
Bye-bye.
Bye, everyone. Thank you so much. Have a good weekend.
Bye, everyone.